Welcome back, students. In this lecture, we'll be covering ion exchange chromatography. Make sure you view the earlier lectures on adsorption chromatography, thin layer chromatography, and paper chromatography. Now, ion exchange chromatography is very interesting and rather easy to understand because most of us have studied purification of water. Okay, over there you already read about ion exchange resins. Okay, the same thing is utilized over here. We need resins for ion exchange, and the first question that will be asked in Viva is, what are the components that can be separated with the help of ion exchange chromatography? So all types of charged molecules. Okay, you are getting it right. Ion means charge, so any type of charged molecules are separated with the help of ion exchange chromatography. They can be proteins, nucleotides, amino acids, so on and so forth. So remember, the solution is to be injected usually as a sample, and the individually separated components are called analytes. Remember, a sample which is a mixture of analytes. So for example, I am giving you an example. Two amino acids. Okay, one is aspartate and another is arginine. So one is aspartic acid and another is arginine. Suppose they are mixed together as a sample. So each of them are known as an analyte. Okay? So we'll be loading this into the ion exchanger. Okay, so how does it act? The basic principle is it's based on the ionic interaction of the analytes along with the stationary phase. So before going to the procedure, let me tell you there are two types of exchangers. Okay, so cation exchange resins and anion exchange resins. These are known. Okay, you already know this from your +2 syllabus of water purification, so you just need to know at least 2 or 3 names of each. Okay, so see, Cefadex gel is one, CM Cefadex, okay, and CM cellulose, these are cation, and DAE cellulose and DAE Cefadex, these are all anion exchangers. Okay, these are commercially made. Now what's the big deal? Anion exchanger stationary phase are the ones that attracts positive, that actually exchanges anions. Okay, and what anions will do? Anions are attracted towards positively charged surface. Okay, the name says if we are dealing with cation exchanger, it means they will exchange the cations. Cations are the positively charged anions and they are attracted towards the negative surface. So just remember this concept. Remember the name of few cation and anion exchanger, and now we will see how this whole thing occurs. The principle is based on their ionic interaction. So see what's going on over here. Cation exchange chromatography retains positively charged cations because the stationary phase displays a negatively charged functional group. Very important. Cation exchange resin has got negatively charged functional group in the stationary phase. You need to remember only one. If you try to remember both and you end up in getting confused, then it's of no use. So remember, cation exchange resins have got a negatively charged functional group. Therefore, cations are more attracted to them. And likewise, anion exchange chromatography retains anions using a positively charged functional group. Now let us take an example. Okay, so the concept is an amino acid mixture at pH 3. Okay, pH being 3, very acidic, is passed through a cation exchange resin. Okay, and the individual amino acid can be you heard that sound, cats are fighting. And individual amino acid can be eluted using buffers at different pH. Suppose a mixture of arginine and aspartate is passed through a cation exchange column. Now ask yourself, you know already the properties of amino acids by now. Which one is acidic amino acid and which one is basic amino acid? Acidic is aspartic acid and arginine is basic amino acid. Now you can calculate which one is positively charged and which one is negatively charged. It's easy. Now, as always, basic amino acids have got extra positive charge. So arginine has got extra positive charge and so adheres to the column. It will strongly bind to the cation exchanger. But negatively charged aspartic acid molecules will not adhere and it will come out first from the column. Simply imagine a column in which cation exchange resins are applied. Now if we give two mixtures, one which has got strong affinity will adhere to the column, and the one which has got no affinity will first come out, and then we need to give elution buffer. In this case, what will be the elution buffer? Weak sodium hydroxide solution. The positive charge of arginine is neutralized. Why? Because sodium has got better replacement capacity. Na plus will replace arginine in the column, thus arginine is eluted finally. The various fractions eluted containing individual amino acids are allowed to react with neon hydrogen agent to form colored complexes. So it's very easy. So remember, in case of cation exchange, we are applying a mixture of different amino acid. The one which has got more positive charge will adhere first, the one which has got negative charge will automatically come out. Then we need to add another more strong cation, which is sodium. So over here sodium will replace the arginine, and then arginine will also come out in the elution buffer. This I have already discussed, the types of anion exchange resins, the cation exchange resins. So you need to remember at least two of them. Now let us just recap what happens irrespective of which is cation or anion. If the molecule is adhered strongly to the exchanger, it will adhere and it will come later. So see, more highly charged molecules are bound more tightly to the resin and slowly travel and are eluted later. So if the attraction is high, it will get eluted later. If it is moderately charged, if the analyte is not a strong anion or a strong cation, it will be separated in between the strong cation and the strong anion. And lastly, the one which carries the same charge as the resin will not adhere and it will come out absolutely at the start. So that's it guys, this is in short about ion exchange chromatography. It's very easy to understand if you have got basic concept of ionic interaction, cations, anions, which one will be adhered first, which one will be adhered later. And again, an example is complex mixture and analyte of proteins and amino acid. And then what will be the final diagnostic test? Diagnostic test will be a color reaction of protein, that is ninhydrin reaction. So pause this video, go back, recap, and then we'll move on to the next phase of the video, which is gel filtration chromatography.